am going to go ahead and start. So welcome everybody to our webinar put on in conjunction with NABT and Bio-Rad Laboratories. My name is Lee Brown. I'm a curriculum and training specialist for Bio-Rad. And all that means is I work with teachers to help them bring labs into their classrooms. So today we're going to be talking about real-time PCR. We'll also talk a little bit about conventional PCR and some experiments that students are doing kind of around the country that you might be able to bring into your own classroom, whether you're doing them as a lab or you're just talking about them with your students and kind of generating ideas and things like that. So like I said, my name's Lee. I've got Tamika here. Tamika's going to be monitoring the chat, and she'll be putting in or interrupting me whenever it's necessary. So if there's some clarifying questions that need to be asked, or if you just come up with a great idea that we need to share, Tamika will stop me and we'll share that. So please feel free to put your questions there. We also thankfully have Rob Park here, who is a scientist at Bio-Rad that works with real-time and digital droplet PCR, and he is going to be handling all the technical questions. So he's one of the experts on at Bio-Rad that can really speak to any of the nitty-gritty details that you are dying to get answered. We also have a few more people on the Bio-Rad educational team in the chat, and they might be popping in and answering questions as well. So just so you know, that's who everyone is. So with that, we're going to go ahead and get started. Let's see here. So today we're going to start off talking about wastewater surveillance. This is something that you're probably familiar with since COVID, but we're going to go a little bit deeper and talk about ways that we're using it in ways that you might not expect, and also the history of that. We'll get into an overview of PCR. That'll be relatively quick. I'm going to assume that most of you probably have some background knowledge on just conventional PCR or endpoint PCR, and we'll talk about qPCR or real-time PCR as well. We'll weave in there, so it's kind of hard to draw dividing lines on this, right? So I want to weave in there how scientists and students can use qPCR to study gene expression. And along that, with that, we'll talk about how experimental design will come into play with this. So the different types of controls that you might need. We've got a good student example that is showing some of their experimental design in here too. And finally, at the end, we'll give you some resources where you can find out more information about all of these things that we're talking about. So real quickly in the chat, pick one of these, rectangles. So where are you with PCR in your course? Are you currently covering PCR, not at all, so you're an A, or are you maybe talking about PCR, but you're not doing any hands-on labs, you're a B. Students might be doing some kind of simulated PCR lab, like maybe they're running gel electrophoresis of simulated real-time PCR or even simulated conventional PCR. So they're doing a hands-on lab, but it's not using a thermocycler. Or D, you're actually doing a PCR lab. So maybe they're looking at GMO, or you've got some independent research going on. Or maybe you have students who are actually doing real-time or quantitative PCR labs, either as part of a course or a lab that you've kind of constructed or led, or independent research that they're working with you or with other outside scientists. So I'm going to give you just a few minutes or seconds here to write your answer here, just so we can get an idea of where you are. It's in the chat. Sorry. So we might have to do this in the Q&A, and I see you all are doing that already. So great. Thank you for that. We run webinars quite a bit, but we usually run them as meetings. So running it as a webinar is a little bit different and tricky for us. So yeah, Q&A is great. Thank you. And I'm just going to pop in there and take a look at kind of what you're answering. See a lot of C's and A's all over. This is great. Okay. So some of you are, maybe looking for ways to talk about PCR with your students. Maybe you don't have a thermocycler. It's kind of a big budgetary thing, right? So it's not insignificant. And so hopefully this will give you some ideas about different things that you can talk about with your students. And for those of you who are already doing, an endpoint PCR or even a qPCR lab, maybe you'll come up with some other ideas here as well. So terrific. I forgot to mention that we are recording this, and we'll send the recording out to everyone that's registered. So no worries there. Okay, excellent. Thank you so much, and feel free to keep popping in the Q&A for that if you'd like. Okay. There we go. So I want to start off talking about wastewater surveillance. And to do this, I want to just start not at the very beginning, but just back at 2005. So think of where you were in 2005. Some of you are probably just, kids maybe. But in 2005, this was the time that the first video was uploaded to YouTube, which is kind of hard to believe. Like I thought it would have been earlier, but 2005, it doesn't seem that long ago to me, but The Office had its first season. It was a total flop. And Hurricane Katrina hit Louisiana and, the Gulf Coast, kind of devastated it. So that's kind of where we are in this world, right? And so a lot of communities around the country were looking at how to monitor illicit drug use. So cocaine use was on the rise worldwide, and your local drug agency is really worried about how it might be affecting, especially young people in urban areas. So how can these social or environmental scientists monitor this really complex issue, right? Traditionally, this was done by different population surveys and interviews. And as you can imagine, this would be a self-reporting situation, right? So there's obvious issues with that, probably not capturing the true level of usage. We also got information from medical records and crime statistics. Crime statistics especially with crime statistics, we're looking at, potentially a good bit of bias there. And so we didn't really have a great way to measure this, right? So in 2005, scientists in Italy were the first ones to develop this technique for using wastewater to monitor the levels, in this case, of cocaine in their city. And those methods were quickly adapted to looking at other illicit drugs, so opiates, amphetamines, et cetera, right? Why is that? Well, urban wastewater is kind of this treasure trove of information. It has all kinds of a mix of the products of human metabolism, so all those metabolites, and those are going to reflect a wide range of factors. So your lifestyle, your diet, whether you're healthy or whether you're infected with some kind of disease organism, right? So a virus or bacteria. And this field as a whole is called wastewater-based epidemiology, or WBE. And that's the chemical or biological analysis and quantification of human biomarkers contained in wastewater, right? There's a lot of benefits that come with using this wastewater-based epidemiology. Obviously, it's anonymous, so we're looking at everyone's poop, not just yours. So this is kind of a part of the ethics are going to come into play here. So depending on how large of a sample size or a pool you're looking at, it can be more or less anonymous. But typically in the U.S., we're looking at very large, like city-based sewage, or maybe from a large university, right? It's non-intrusive. No one needs to go in and get a test. It's relatively inexpensive. So, we'll go through that in a little bit compared to other methods, especially. And it's relatively quick. We can take samples at least once a day, if not more often. So, and get some good indicators of what kinds of things are in the wastewater there. This wasn't the first time wastewater was used to gather epidemiological information. So way back in 1939, and this was from 1940, so this is a follow-up paper, scientists isolated viable poliovirus in sewage, right? And so they reinfected monkeys with that. Monkeys sadly died. And so they realized that there were viruses that were shed into the sewage system. And, you can go even farther back than that to physician John Snow's finding in 1850 that cholera outbreaks in London were connected to contaminated water. So this WBE, wastewater-based epidemiology, is still used today to monitor polio. And you may remember in July of 2022, a case of paralytic poliomyelitis was confirmed in an unvaccinated adult in Rockland County, New York. So they found that when they sampled the wastewater, that they found evidence of poliovirus transmission in the wastewater. So obviously we've come a long way from, isolating polio from wastewater and infecting monkeys. We were having to look at whether the virus was viable or not by reinfection, or, doing plaque assays at some point. And now we're looking at techniques, we're using techniques like qPCR, right? So this was a pretty limited field for decades, and it typically focused on tracking specific health threats in specific locations. So in Israel, they looked for polio. In Arizona, they looked for opioids. And Vibrio cholera in Peru, this is one of the papers from that. Okay. And then in 2020, we had, kind of this sea change in how everything happened with research and environmental surveillance, right, when COVID hit. And so in 2020, COVID came along. There was an explosion of scientists and papers related to using this environmental surveillance, and in particular, wastewater based epidemiology, to see what viruses and other disease-causing organisms were in our sewage. So scientists started monitoring our collective poop, and we looked to see where levels of SARS-CoV-2 were rising. Since then, we've seen kind of this We've seen this explosion of the number of scientists in this field, right? So it went from just dozens to tens of thousands that are currently doing this kind of research. And the CDC launched the National Wastewater Surveillance System, or NWSS, in the fall of 2020. And even more testing programs have launched worldwide. And so that's what you're looking at here is this COVID Poops 19 dashboard run by the researchers at the University of California in Merced, right? And so this is, I think, current from today. So we're looking at 72 countries, and you can see there's airports, there's universities, there's all these different testing sites, and they're all kind of compiled on this dashboard here, which is really cool. If you're interested in looking at something close to you on your own hometown, you can just Google this. A lot of this is there's dashboards still up for large cities, especially. So I'm in New Braunfels, Texas, which is kind of central Texas. The closest one to me is the Houston wastewater monitoring dashboard. I'm sure San Antonio probably has one as well. I just didn't happen to find it when I was researching this. But this is a really nice one from Houston. And I remembered back in July in Texas, it's been just terrible. So we had a terrible drought, and temperatures over 100 for it felt like forever, but it was a good solid 2 to 3 months. Everyone was inside, and as we've seen the past 3 years, whenever people are gathered inside with less ventilation, less airflow, that kind of thing, COVID levels go up. And so that's what you're looking at here. This is from July, and you're looking at a 927% increase in viral load comparison to July of 2020. The positivity rate was 31%. Right? So very high. And so this kind of information really allows epidemiologists to get a heads up. Oftentimes, especially now, we're not seeing people going into clinics to get tested. We're doing that at home, right? And so they can kind of get a heads up on what's going on locally. Is there a spike in COVID cases? Are we at a relatively, lower time or not? So how does that happen? Well, there are actually little, robotic sample takers, and that's not the right word, but I'm drawing a blank here, that will go and collect samples from the wastewater treatment plants. These are, I've heard them described as the size of like R2-D2, which gives me a good idea of what that might look like, right? And so they're going and capturing a sample of wastewater. They might do this every day, every X number of hours. Obviously, for monitoring, the virus needs to be shed in either feces or urine, right? And present in sufficiently high levels. So you're looking at here an example with COVID, but this could really be applied to a number of things, and we're going to talk about a lot of those today. So they get the sample, they're going to centrifuge it, and depending on what virus or what they're looking for in the wastewater, sometimes they'll use the liquid from that sample, and sometimes they'll use the solids from that sample. So polio, I believe, is typically they're looking at the liquid from these samples, and with SARS-CoV-2, they found it was better to look at the solids. They take that sample, they lyse it. In the case of COVID, we're looking at RNA, so they want to extract that RNA, make sure it's good and intact. And then PCR, as you probably know, only works with DNA. So for the case of COVID, we're going to need to take that RNA and make it into cDNA in a process called reverse transcription PCR. So we'll talk more about that in a little bit. Then they'll throw that into a thermocycler and look and see what we've got, right? Okay. I'm going to pause here and just make sure there's no questions or anything like that. Okay, so using that basic technique, there is all kinds of things that we can actually monitor in wastewater. So flu, newly emerging diseases like mpox, formerly called monkeypox, drug-resistant pathogens like Candida auris. And so, here's just a list of just a few of them, right? So there's this could be slides and slides full of things here. But microbial indicators that might serve as proxies for some kind of fecal contamination. We could look for this, not, obviously not in wastewater because it's clearly contaminated with fecal matter, but in rivers and streams, things like that. We can also look for pathogens like norovirus, different bacteria, giardia, things like that, looking at waterborne diseases. Antibiotic resistance genes, this is a huge field right now, looking at, they're doing this around different farms and things like that, to track antibiotic resistance levels in the environment, and genetic markers for heavy metals, algae blooms, looking at genetic materials, so the specific species that might be harmful, and tracking endangered species as well. So they can look and see if those endangered species are there, how, are their populations declining, are they increasing in a specific area. So lots of good ideas here. And, we've seen students and teachers go after a number of these, ideas. So I'll bring up a few later on, but hopefully this kind of gets your creative juices flowing. So let's real quickly, or not real quickly, we won't go too fast here, but let's go over PCR and qPCR, right? And so, back in 1983, we have Kerry Mullis here. He was a scientist at Cetus Corporation in California, and he came up with a way to amplify copies of DNA using a DNA polymerase and primers that bound to each side of the DNA sequence that you wanted to amplify, right? And so with repeated cycles of heating and cooling that allowed the primers to bind and the polymerase to add nucleotides, you could copy the DNA over and over, essentially DNA replication in a test tube, right? In the middle here, we have one of the earliest automated PCR systems. And I think, I have heard rumor of the fact that in some schools, teachers do this kind of thing where they have just 3 water baths. So we need 3 different temperatures, excuse me, 3 different temperatures usually for conventional PCR. And this is just an automated version of that. So it's, the 3 different temperatures, taking the tube, moving them to the different water baths. Before tech polymerase was, thought of as a way to have a heat stable polymerase, you'd have to add a polymerase at the end of every cycle. And on the right here, we have Mr. Cycle. So this is the first prototype for an automated PCR machine from Cetus. And, if you look real closely, there's like this California Dreaming sticker on the side. I love that. Maybe some kind of homage to Mullis. So this is in the Smithsonian, which I think is super cool. I will have to visit that the next time I go. And here's where we are today. Obviously, these are Bio-Rad thermal cyclers. So we have on the left there, we have a PTC Tempo. That's one of our newest, just endpoint conventional PCR or thermal cyclers. In the middle, we have a CFX Opus, and that is a real-time PCR machine. And on the right, we have a digital droplet. This is kind of top of the line. I'm not going to talk a lot about digital droplet here, but we have a whole webinar coming up where we're really going into detail on that. And that's kind of where the field, especially of wastewater monitoring, is going. So I'll put that information up at the very end. So we've got, we've come a long way in not very much time, right? This started in the early 80s. And so we've really, made some major strides. So ever since PCR was invented, it's been kind of this cornerstone of molecular biology in basic research, in biotech, medicine, forensics, diagnostics. Any application that relies on DNA will almost certainly use PCR at some point along the way, right? In this presentation, we're mostly going to focus on quantitative or real-time PCR over here on the right. But first, I just want to start with a quick review of conventional or endpoint PCR. And nomenclature is kind of a big deal with PCR. So I apologize in advance for going back and forth. I think most people, when they say PCR, at least in the education world, are probably just talking about endpoint PCR, conventional PCR, where you throw your things in a thermal cycle, throw your DNA and and polymerases in a thermal cycler, let it amplify for 40 cycles, and then run a gel at the end, right? And so that is a qualitative analysis. Is my target DNA present or not? So we'll either have a band on the DNA or on the gel, or we won't have any band there, right? Yes or no answer. For other applications, scientists may want to know not only if their target DNA is present, but how much is there. So for that, they're going to need to get a quantitative assessment for how much target DNA is present. And for that, we'll need to use quantitative PCR. So that quantitative PCR, you might have heard referred to as real-time PCR, sometimes referred to as RT-PCR, which gets really confusing. I have a whole slide on that coming up. Or qPCR, which is what I'm going to refer to it as through most of this presentation. Okay, so we're going to start off with conventional PCR. So conventional endpoint PCR, you can see there's a gel here at the top right. This is actually from a workshop that we did a while ago with teachers were isolating DNA from different plants to see if they had isolated DNA with plant genes and with GMO genes, right? So I won't go into details on that, but just know that's what you're looking at here. These bands up here show that they successfully isolated plant DNA, and this one is showing that this particular test food had DNA with GMO sequences in it, okay? And so with conventional endpoint PCR, we're amplifying a specific sequence of DNA, looking at the result using gel electrophoresis at the end. And we still use this a lot, right? This is not something that is completely replaced by real-time PCR. Oftentimes, we just need to make a bunch of DNA, right? And so we might need to use that in downstream applications like sequencing or cloning. It is relatively affordable, and we have students all over the country, even at the high school. I even have middle school students doing this at a school in Houston and looking at DNA in their own cheek cells, as well as this GMO lab here. Okay, so quick review of what do you need for PCR. Typically, you'll have a PCR master mix. Sometimes this is called a super mix. That will include a DNA polymerase, right? So that's what's going to add nucleotides to our template. You'll have these DNTPs, so GAs, Ts, and Cs, some buffers. You'll have magnesium chloride, and all that together is typically in a tube of master mix or super mix. To that, you'll add your template DNA, so that's the DNA that you want to make more copies of, and your primers. Those primers are going to bind at the flanking regions of the template DNA. Throw all that in a tube, add that into your thermal cycler, program this. This is really easy to program typically. Just I'll show you kind of what a typical cycle looks like. And these repeated heatings and coolings will allow the polymerase to make more copies of DNA. So first of all, let's take a look at how that looks. So we have our template DNA here, and we're going to increase the temperature up in the 90-95 region to denature that template. So the polymerase needs to get in there, and it can't do that unless the two strands of DNA come apart. So we have denaturation. And then the temperature comes down a little bit. So at 60 degrees, thereabouts, these temperatures are going to depend a lot on your primers and how much GC content. There's a whole, there's whole courses on primer design and things like that, but these are just rough guidelines. So lower the temperature a little bit, the primers will come on and bind. You can see there's forward and reverse primers, since that polymerase only goes in one direction and we want to copy both strands. So those primers will come on and bind, and then we'll crank up the temperature just a little bit. This is the temperature that Taq polymerase likes to work at. And so Taq polymerase will get on and add all those DNTPs along that strand and synthesize a new strand of DNA. Okay, and so here's kind of that whole process, looking at that all at once. And in theory, we're going to get a doubling every cycle, right? And so if you look at the left, we have our starting DNA, which is, kind of this light blue and dark blue. That's just, a representation. Obviously, DNA is not colored like this. The second cycle, we have 2 copies of that. For every 1 copy of our starting template DNA, we're going to get 2 copies on that second cycle. On the third cycle, we'll get 4. On the fourth cycle, we'll get 8. And so if you look at Cycle number 3, here's really the first time in the middle here. Oh, hold on, I forgot that I'm on my other screen. This guy, here's our 2 complete newly synthesized strands of DNA, right here. These still have a strand of the original starting template DNA. And then by cycle 4, we have even more of those. Okay, and of course, this goes on for 35 to 40 cycles. So at the end of that, for every 1 DNA molecule we have, at the end, we're going to have millions of DNA molecules, right? And so here's kind of what that looks like. So we have in cycle 1, from our 1 template, we're going to get 2 new pieces of DNA. In cycle 2, we're going to have 4 copies. I'm sorry, I don't know why I said pieces, copies of DNA. In cycle 2, we'll have 4. In cycle 3, we'll have 8. Cycle 4, we'll have 16, on and on and on. And so we kind of get this theoretical doubling. Now, that's not exactly right, because the conditions become less favorable over time. And so the efficiency of the amplification is going to decrease. So we're running out of DNTPs, our buffers might be getting a little bit unstable. The reaction dynamics just aren't as favorable anymore. And so we'll tend to see this tapering off of the template in later cycles. This isn't something that you would necessarily see or notice with conventional PCR because we're just looking at the very end, right? So we're just seeing how much we got at the very end. Oops. Okay. And for conventional PCR, the detection of the PCR reaction occurs at the end. So once your thermal cycler is done running, this might be 2 to 3 hours later, typically, you're going to take samples out, add a stain or loading dye, and then stain it afterwards, run a gel, and see what you've got. So you can get like I said, you can get a qualitative answer here. Yes, I have DNA. No, I don't. It's really hard to say how much though, because we're not looking at the starting material here. This is not controlled in that manner, right? Okay. So now let's take a look at qPCR. So the key ideas we're going to look at to detect a specific target of interest. And we're going to do that by amplifying our region of interest in the DNA sample. And we're also going to monitor that over the course of the reaction. So not just at the end, but after every single cycle, we're going to basically take a picture of things and see what we've got, right? So that's a little simplistic, but that's kind of the basics of it there. So real-time PCR gets its name because instead of running to an endpoint and then running a gel, the detection of that DNA is performed on the machine itself, right? On the real-time instrument after each PCR cycle. The DNA isn't directly visible to the real-time PCR system, but instead relies on fluorescent markers, and we'll talk about that in a minute. And so the machine is looking at a relative amount of DNA present in the sample in real time. That's where real-time PCR gets its name. So as the reaction progresses and you get more DNA produced, the fluorescent marker becomes bound to the DNA, resulting in this increase and in this amplification curve that you can see here. So by incorporating these fluorescent DNA markers, probes or cyber, things like that, into the reaction mix, we can detect the relative amount of nucleic acid present in the reaction in real time. And we can do that by doing running that with a reference gene. Okay, so after every cycle of amplification, the fluorescence is of a specific wavelength is emitted by an LED housed inside of this optical module, right? So on the top of this machine here, it's absorbed by a DNA-specific fluorescent marker inside the reaction, and that is going to just re-emit that energy at a different wavelength that's then detected by the system. Let's take a little bit of a closer look at this, okay? So in 93, Professor Russell Higuchi described real-time PCR, and again, using these fluorescent detection strategies to monitor amplification. Typically, you're going to start off with a dilution series of a known template to use that as a quantitative reference. And so this is a really good way for students to start. What we're looking at here is tenfold dilutions. And so I'll get more into this amplification curves later, but I think you could probably see here that each of these dilutions, so this is the most concentrated to the least concentrated over here. The most concentrated, if we look here, it looks like it was done with 3 or 4 replicates. So if you're looking to really test your students on their pipetting, qPCR is a great way to do that. So they have to be really, really precise with their pipetting to get these really nice trace lines here, right, that are overlapping like that, and to get them spaced pretty evenly. Okay. The more cycles it takes to reach a certain fluorescence, the lower the amount of initial target molecule, right? And so let me say that again. So over here, like I said, on the far left, we have the most sample, right? The most concentrated sample. And so we're going to see that fluorescence coming up sooner than we are over here on the far right, where we have less sample. So the fluorescence is happening, we're starting to see that on the machine after 32 cycles or 30, 31 cycles versus, after 10 cycles with our most concentrated sample. Okay. There are 2 main fluorescent chemistries used in real-time PCR. Dye-based chemistry, typically main example is cyber green, and probe-based chemistry. And we're going to look at both of those. So cyber green is an intercalating agent. It's going to fit into the minor groove of double-stranded DNA. So you might be familiar with ethidium bromide, similar to that. When it binds to double-stranded DNA, the fluorescence increases 1,000-fold, and so it's readily detected by the real-time instrument. And so here's kind of what that might look like if we're, you can think back to that previous slide with conventional PCR. So with, our template, cyber is kind of non-discriminatory. It's going to bind to any double-strand DNA. So even with our starting DNA, we're going to have cyber binding here and fluorescing. But there's so little of our DNA template at this point that the real-time system isn't going to pick that up, right? And so again, we have denaturation at a higher temperature, 95 degrees. And so we have those, the template strands coming apart. The fluorophore is no longer fluorescing anymore because there's no more double-strand DNA in this particular piece right here. The temperature goes down, primers are annealed, are binding, and then raise the temperature back up. So we have extension. And as soon as we get that double-strand DNA, again, cyber will bind and cause that fluorescence. So the key concept here is basically the higher the concentration of double-strand DNA you have, the higher intensity of fluorescent signal you have, right? And so again, more double-strand DNA means more of this fluorescent signal. And you can kind of see that over here on the right. So we don't have any application. This might be a control that you have. Instead of a template, you just have water. So a no template control in there. There's not going to be any amplification because we don't have any double-stranded DNA in that sample, right? And in our target DNA or our target sample right here, we are amplifying that over time. And so we see more fluorescence over time. So down here, we have fluorescence, but it might be below the threshold of what the machine can see.
All right, Lee, if I can interrupt you for a second, we have a question in the chat.
Yeah.
Robert asks, what about quenching fluorescence? How does that affect the signal?
We'll talk about that. Good question. We'll talk about that with probe-based chemistry. Is that what you said, quenching fluorescence?
Yes.
Okay, yeah.
Thank you.
Hold tight, Robert. Good question. All right. Okay. So let's take a deeper look at these amplification conditions. The 40 cycles happen, right, of denaturing and annealing. And, I want to point out here that we're looking at, instead of 3 temperatures that we were looking at before, even on the previous screen, oftentimes you can just do 2 temperatures, that 95 degrees at higher temperature to denature the DNA, and then somewhere in the 55 to 65 range is where your primers can bind and extension can happen. So you don't necessarily have to go up to 72 like we were looking at before, right? So if you're, this is often, I think always probably preceded by a 95-degree hold, where a polymerase blocking agent will break down and activate the polymerase, right? And so that way, all your amplification starts in one time and all of your wells at the same time. And it also increases the specificity of the amplification, I'm sorry. Dyes like cyber green are non-specific, and so they'll bind with high affinity to anything that's double-stranded. And so, you would need to verify that the signal that you're detecting is specific to the target DNA, right? So we are going to do a melt curve analysis after this. So after those 40 cycles of amplification, the temperature will be increased stepwise from 65 to 95 degrees. Energy is required to break the hydrogen bonds of double-stranded DNA. And so a melting temperature is a characteristic of a double-stranded DNA fragment that's based on the GC content and the length of the amplicon. Okay, so after we amplify our samples, we're going to heat them up to ensure that we have a single homogeneous population of amplicons. We don't want several different peaks at this point. Okay, so there's that melt curve. And again, this is just at the very end of the 40 cycles. And so what we're looking at here is a typical melt curve. The fluorescence intensity is plotted on the y-axis against the temperature on the x-axis. And so we can see here, as the temperature goes up, our fluorescence intensity goes down. And let me click on this here. The melting temperature is indicated by the arrow here at this point of inflection. And that is indicative at the temperature at which half of the DNA is double-stranded and half of it is single-stranded. And so if we look at the negative derivative of the rate of change here of fluorescence versus temperature, this is something that is done all in the software that comes with the thermal cycler. You're not going to have to do the math on this. We can better visualize that inflection point. And as you can see right here, we have one nice inflection point instead of multiple ones, right? So if we, for example, had a primer dimers, we would expect to see those kind of at the left end here coming apart at lower temperatures. And so, you might need to, you would need to do this to confirm that you're only amplifying one product, and you might need to tweak your parameters a little bit if you're seeing multiple peaks here. Okay, now to Robert's question, probe-based detection. So the other main type of dye-based chemistry, or of chemistry that we're looking at with real-time PCR, is probe-based detection. So these are probes that are synthesized that will bind to your template DNA. The probe is going to sit in between the forward and reverse primer, and it's typically 20 to 30 nucleotides. At the 5' end, it's got a fluorescent reporter dye. It might be FAM or hex, something like that. The 3' end will be conjugated to a quencher molecule. And so when this probe is not bound to a DNA template, the fluorescence isn't detected because that that quencher is there, right? So the energy absorbed by it's not freely emitted because it's located close to the quencher. So now let's take a look at, during amplification, what happens when the polymerase kind of hydrolyzes that probe. It's going to break free break free the reporter, the fluorophore there, from the quencher, and then that will be releases fluorescence that can be detected by the real-time system. Okay. So again, here we have our initiation, denaturation. We have the primer annealing. Here's our probe right here, right? And so this fluorophore is quenched by the quencher right here. Once DNA polymerase comes along, that will be hydrolyzed. So we'll cut that, and that fluorescent reporter will be released. And that is what the real-time system is going to detect now. Okay, so that fluorescent signal is only detected once that probe is hydrolyzed from the template. Okay. Any questions on this? It's a little bit, this is kind of a lot, so I'll pause for just a minute. And again, same end result, that the more DNA copies we have, the more of these fluorescent reporters are going to be released, right? So we'll see a similar amplification curve. The nice thing about using these probe-based chemistries is that they can be multiplexed. And so you can have different fluorophores and create those probes that are specific to different targets of interest. So depending on your machine, right? So here's one looking at 5 different channels that you could look at and detect and multiplex, looking for different targets, right? With the probe based. So kind of an overall big picture here. So cyber is, pretty inexpensive, comparatively inexpensive. You can run that melt curve analysis to make sure that you just have your single target that's being amplified. You can't multiplex it though, right? Because it's going to indiscriminately bind to any double-stranded DNA. With the probe-based chemistries, you can multiplex. You're going to just put a different fluorophore here based on, depending on, on different oligos there. It's more specific. You're creating that oligo based on your target molecule. And as you might imagine, these are more expensive and they're, a little bit more complex of a design. At the end, you're going to need to run an agarose gel to confirm the specificity here. So you can't do a melt curve on this one. Okay. So now let's take a look at gene expression. So, and I want to back up a little bit here and briefly talk about this nomenclature, RT-PCR, which I'm sure you've talked or heard about. So what are we talking about? Oftentimes we're talking about real-time PCR, right? So we just talked about this. We have our cyber and our probe-based chemistries here that will bind to DNA, emit this fluorescent readout here, and our machine will measure that, right? And so we can get this amplification curve here. But there is also another type of RT-PCR. So let's say that we're looking at, I hate to bring it up again, COVID, right? SARS-CoV-2, it's an RNA virus, or maybe we're just looking at gene expression. We're doing a gene expression study and we're trying to look at the mRNA in our sample, right? So if we're trying to look at if our starting material, if our starting genetic material isn't DNA, but it's RNA, we need to do something called reverse transcription to take that RNA template and make it into complementary DNA. And that process is called reverse transcriptase PCR. So that is also abbreviated as RT-PCR. So you might have RT-PCR, right? And so whenever you see RT-PCR, someone's talking about it, you might want to just make sure that in your mind, you know what they're talking about, or you might want to get some clarification there, because both of these are very commonly used abbreviations for both real-time PCR and reverse transcriptase PCR. Because of this, I think most people, and I know for sure at Bio-Rad, we're moving towards using qPCR instead of real-time PCR, but old habits die hard, and so you'll still hear real-time machines, real-time thermal cyclers, real-time PCR instead of quantitative PCR. But the reason for that movement is to do some disambiguation here. Okay. So let's take a look at an example student investigation here. And I can link this. I just took some screenshots of her, she has a really nice PDF here. But the student was looking at the relationships between FOXO3 gene expression and collagen expression levels. Why? She was interested in ACL injuries. Which she was looking to see if there was some relationship between this FOXO3 expression and collagen, either if those were related and how those might tie into ACL injuries. And so when you're doing a gene expression study, the first thing that you're going to do is isolate your RNA. And there's lots and lots of factors that come into play here on how to get a good quality of RNA. So RNA, if you look at it wrong, it will degrade. So there's lots of ways to get those out of the cells, and they're stable and not impure. Then they'll take that RNA, do reverse transcription to get your cDNA. You'll need to set up your qPCR and figure out how to, sorry, I've got a typo in there, figure out how that assay is going to go, do the experiment, and then analyze your data, right? So that's the big picture here. So again, I pulled these from her PowerPoint here, but looking at the relationship between this gene FOXO3 and collagen, again, she's looking to see, is there some relationship between the level of FOXO3 expression and the genes involved in collagen synthesis, okay? So on the left, you can see, the background on collagen, so that COL1A1 and COL3A1 are important genes in collagen, and FOXO3 is a regulatory gene involved in apoptosis and inflammation, right? Also lengthens telomeres. So typically now what's going to need to happen is when you're doing these gene expression studies, again, from the cell, you can do this reverse transcriptase PCR. Two different ways to do it here. In this example, we're looking at a two-step method where you take your RNA, you have your RNA isolated from the cell, right? So here's our cell, here's our RNA down here. And we're going to throw in these reverse transcriptase. These are typically from viruses, our dNTPs, our oligo-DTs, and do reverse transcription. And that will generate a cDNA, a complementary DNA template. Then we can take that and do qPCR with that and amplify it, okay? The nice thing about doing the two-step method is you can take the cDNA and stick it away in the freezer for analysis later on, right? And so, some researchers choose to do it because of that. You can also amplify it multiple times here. Okay, and this is the method that this young lady used when she was doing it. She did mention that she needed to do a couple of rounds of amplification. Okay, so here's what her investigation looked like. She had access to the ligament tissue from knee cadavers and isolated that messenger RNA, and then did reverse transcriptase to create the cDNA. Just as an aside, this can also be a one-step process. Bio-Rad sells a kit for this too, where you can put everything, the reverse transcriptase and the DNA polymerase, along with gene-specific primers together, all in the same tube, instead of doing that two-step method, and do that all together, right? And so the upside of this is, of course, you're less likely to contaminate samples. It's faster. The downside is that you don't have that cDNA that you can kind of separate out and use again later, right? Okay, so what time is it? 6:20. I'm going to go through this a little bit more quickly just because I want to get time. I want to leave at least 5 minutes at the end for questions. And I do want to tell you, though, that we have a whole, oh gosh, I think it's 5 or 6, it's a whole course on this stuff that goes into way more detail than I can in just an hour. So if there's any questions at the end, just know that you can refer to that as well. So we have that available online. I'll show you how to get there, and we have webinars coming up as well. Okay, so we've already actually looked at this, so I'm going to skip ahead. Okay, so here is our amplification curve, and you'll see that we have an initiation phase, an exponential phase, and a plateau phase. This threshold will be drawn kind of where we start to see this exponential phase going up, and wherever this line crosses our cycle number, or wherever we have our curve crossing the threshold, we'll draw a line down there, and that CQ value will be the cycle number down here. Okay, that's a cycle quantification. Okay, so we've looked at, amplification. We've looked at how we're going to detect it. Now let's look at what the machine actually spits out at the end, right? So typically amplification is going to be around 40 cycles of this heating and cooling. And at the end of that, the level of fluorescence is going to be measured and recorded for each of your experimental reaction wells, right? So these are typically done on a plate rather than just the individual PCR tubes. And so during the initial cycles of the amplification, the fluorescence is going to be really, really low. You'll have it there, let's say we're using cyber. Your template is going to have cyber bound to it, and there will be fluorescence. It's just that your machine will not be able to detect that, right? So it'll just kind of be this background level of fluorescence. And the curve is set to take off. Let me see, I think I've got this animated. There we go. At the point that the level of the fluorescence kind of moves away from this background level. So it's moving away from the background fluorescence. Okay. There we go. And the software on the real-time system is going to draw the smooth line through each of the data points. So again, if we're looking back here, these are the data points. This is kind of, the quote unquote picture that's taken at the end of every cycle. So your machine will then draw this smooth line. That's the amplification curve. And so for cyber green or a singleplex probe-based assay, so one that you haven't multiplexed, right? Each experimental reaction well will have a single amplification curve. If you're doing multiplex PCR, you will have multiple amplification curves, right? And typically those are denoted by different colors, okay? There's 4 real identifiable parts to that amplification curve. Broadly, we'll have the exponential phase and the non-exponential phase. And I'm just going to go through these pretty quickly. Like I said, I want to make sure we get through. But at the beginning, we'll have kind of this baseline where we have amplification starting. The fluorescence is really too low to be detected. That takeoff where we're really getting that exponential increase is starting. This is where you're going to see your best amplification efficiency, the best precision. This is typically where you want to set the threshold. The machine will set the threshold, but where you would set the threshold is somewhere in this phase. Okay, and then we have the linear stage. So at this point, your PCR reagents are starting to decrease. So you're running out of stuff, not too much, but as you go, the more things you'll run out of. Your amplification efficiency will go down. We're having a little bit less precision, but we still have that linear increase. Okay. And the plateau. So at this point, the amplification is going to flatten off, and the amplification efficiency is greatly decreased, and there's going to be minimal detectable amplification, right? This is mostly due to the depletion. You're basically running out of reagents here. You're also going to see some self-annealing of the PCR products, okay? This typically happens after cycle 30. And so it's not usually advised to go beyond 40 cycles of amplification. If you do that, you're going to increase the likelihood of getting non-specific amplification. I'm going to skip here and go to experimental design. So, and I actually want to just go ahead and look at the design of our student here. So when you're looking at experimental design, you want to, what is your question here? What are you measuring? What are you trying to measure? And how are you going to set up your experiment? And so with our student, our example from earlier with that FOXO3, she was looking at whether or not the FOXO3 gene expression and one of these collagen gene expression levels were correlated. Is there a direct correlation? Either negative, or is there no correlation, right? And is there a relationship between that gene expression level and ACL injuries? Okay, oops, sorry. The other main thing that you need to consider with your experimental design, and there's lots of things here, but I do want to talk about controls. So you need your controls, here's just another random gene expression study. So this is maybe a researcher looking at the expression of a particular gene after some treatment in mice, right? And so the first one is, of course, having different biological replicates. After you extract the RNA from your sample, in the case of our student, it would be from the knee, the cells in the knee, the cadaver knee. In this one, it would be from the mice. So I think this was like a liver experiment, right? So there's controls that you want to look at here. How good was your RNA quality and how much of it did you get out? Then you do reverse transcription. And again, you're going to need a control here. So a sample that does not, that you did not do reverse transcription on. And finally, amplification. So once you put it in the thermocycler. And so there's lots of things to consider here. Again, this is really, we go into a lot more detail on this in our course. What I do want to mention real quickly is just reference genes. So we want to use reference genes to, and these are kind of, I guess, when I first learned all this is called housekeeping genes, and that nomenclature has kind of fallen out of favor. But reference genes are what we use to normalize the signal between samples. So we want to control for the amount of starting sample, and we want to make sure that that reference gene is stably expressed across all of our sample conditions, right? In the perfect world, you're going to be able to use multiple reference genes. And I know in our CFX, thermocyclers, we have, you can get different plates that have reference genes on them, and it will tell you which one to use, right? So you can run those kind of as your control along with your samples in there, and it will tell you which reference genes are best to use. Looking back at our student's reference genes here, so she used a gene called HPRT, and she wants to look at the change in the CT. CT and CQ are interchangeable. So CT is for cycle threshold, C2, it's, like I said, interchangeable with CQ. So a change in those values to determine if that gene expression is increased or decreased or stays the same. So let's take a look at just a generic reference gene here. So here's our target gene of interest that we're looking at. A and B, right? And if we look here, our target gene appears to have, this threefold increase over condition A, or in condition B as compared to condition A. But then let's take a look at our reference gene, right? So here's our reference gene, and we also have a threefold change in our reference gene between conditions A and B. So that's going to tell us that, that's either due to unequal sample loading. It could be just some kind of transcriptional noise, but that threefold change is probably not particular to just our target gene. So if we looked at the normalized gene expression, we would see no change, right? So this is kind of why reference genes are important. I'm going to come back to that. Oh, dang it, don't I have hers? So I don't know if I'll have time to get to hers, but what she found was, even though she did see I've got to see if I've got that. Let me see if I can find hers before we're done here. I want to get to the end. Oops. Dadgum. I'm going to go over here. Oh, I don't have it. I'll stick this in here and send this out to you. What she found at the end of her experiment, the first time she ran this, it looked like there was some correlation between those FOXO3 genes and the collagen genes. However, when she ran it with the housekeeping gene, or with the reference gene, I'm sorry, she could see that those effects just kind of went away. And so, not what the not what she may have wanted to happen. That's kind of a little bit of a disappointment, I'm sure, but a good learning experience for her as well. So real briefly, I have just a couple more slides here. Let's see if this will work here. Is it still doing this? Sorry, no, it's not. Hang on, I'll show you on here. So we've got a couple more examples. River City High School, this is in West Sacramento. They did coliform monitoring at the Sacramento River. They filtered the water, extracted the DNA. So they didn't need to use reverse transcriptase here, and then performed qPCR to assess the concentration of coliforms in that water, right? So pretty cool stuff. They're no longer doing this as far as I know, but they are still using their instrument for other experiments. A school in Hawaii was using qPCR to monitor one of their local waterways for flesh-eating bacteria, and this flesh-eating bacteria actually led to deaths in the canal that runs by the school. And so they did this for several years, monitoring different species of Vibrio in the canal. Okay, so pretty cool stuff. We've got some other resources at the end here. Bio-Rad has, I'm part of the education team, and we have a bunch of really cool kits for PCR, whether you have a machine or not. So if you're just going to talk about PCR with your students, we have some simulated qPCR ones that are just running a gel. looking at opioid detection, and viral transmission is one of our newer ones. We have endpoint PCR kits, and we have kits for using real-time or quantitative PCR as well. Okay. The academy.biorad.com is where you can take a whole course on this stuff, as well as other, if you're interested in Western blotting or digital droplet, we have that as well. We have thermal cyclers for all your needs. I'm not going to go into those much here, but I do want to make sure that you know that we have more webinars coming up. And November 3rd, we're going to have a scientist come and talk to us about his water quality research that he's doing. And then on November 17th, we have another scientist talking to us about viral vector development at her company as well. Both of these are using both qPCR and digital droplet PCR, which I didn't have time to get to in this one. If you're really interested in doing a deep dive, Innovate Bio is sponsoring all of these, but we're working in partnership with them and St. Louis Community College. There will be an advanced PCR workshop this summer that is in person, week-long, where you will do real-time PCR and digital droplet PCR and kind of get some more hands-on knowledge of these subjects. So thank you so much for attending. I am going to stick around for questions, and I think we might have a few other people that can as well. If you need to drop off, please go ahead and do so. And again, hope to see you at another webinar or in person soon. Thank you so much.